I am really excited to tell you about what we are doing, and not only that, I'm going to tell you about what we're doing, but I'm really looking forward to talking to you all and getting feedback and questions at the end. So I start by introducing you to Haven. Haven was born at 24 weeks during pregnancy, when a normal pregnancy is 40 weeks. He was less than a pound when he was born, and I was there to lead the team who took care of him in the first 2 weeks of life. As you can see, Haven is very small, and during his 5 months of hospitalization, he had respiratory failure. You can see that he has a breathing tube in his mouth because he cannot breathe by himself. He had hemodynamic instability. He needed all kind of help with maintaining his blood pressures. He had multiple intestinal surgeries because as soon as we started feeding him, his intestines perforated. And he also went through multiple infections, which all of these complications are really not uncommon for extremely preterm babies. And by extremely preterm babies, I mean babies who are born less than 28 weeks of gestation. He was really born around, like, at the edge of viability. So what all of these insults or things that he went through have in common are episodes of systemic hypoxia and inflammation. And from what we know, hypoxia and inflammation are the major environmental risk factors that are associated with neurodevelopmental injury in babies who are born preterm, and specifically extremely preterm. So when my lab is focusing on understanding the developmental hypoxic brain injury, and Haven and preterm babies are one of the patient population that we are very interested in understanding, because to be honest, we have no therapies for them. The other injury that is highly important, and Evan is working on this, is hypoxic ischemic encephalopathy. And these are injuries and hypoxic injuries that happen in term babies around the time of birth, like the placenta gets detached, or the cord gets tangled, or just cannot really transfer oxygen and blood to the baby. And then also congenital heart disease. Babies with congenital heart disease, some of them, even at the time of birth, because of all the mixing in the heart, they have mixing of the blood in the heart, they have lower brain volumes, which is very, very interesting. So when you think about development, it's really not surprising that both preterm babies and term babies who suffer from hypoxic events are at risk for neurodevelopmental impairments. If you look at around the 24 weeks post-conceptional week or gestational week, you see that a lot of things are still happening in the brain. There is still neuronal migration, neuronal differentiation and formation of the cortical layers, and neurogenesis is still ongoing in various parts of the brain. Synaptogenesis has barely started, and gliogenesis is also barely starting. At 40 weeks conception, which is the equivalent of term birth, again, multiple processes are affected, can be affected. At this time, you already see synapse refinement. There's still some neurogenesis, there is synaptogenesis, and then also the gliogenesis is really kind of at its peak. So with that in mind, in the past 20, 30 years, we learned a lot about the risk factors associated with neurodevelopmental impairments. We know what the neurodevelopmental outcomes are because of very good longitudinal clinical studies. We know about the anatomic abnormalities because of our radiology and pathology colleagues. However, we still have a limited understanding of what is happening at the cellular level, particularly in the human brain. And as a result, we have no biomarkers of injury or recovery for these extremely preterm babies, and then or HIE or congenital heart disease babies, and we also don't have effective or targeted clinical interventions. And a lot of the neonatologists are really trying to figure out what we can do, because prematurity, for example, and encephalopathy of prematurity, how we call it, is the major risk, is the main cause for neurodevelopmental impairments in children. So keeping in mind that essentially all models are wrong, but some are useful, in my lab, we have focused on developing human models for developmental hypoxic brain injury. And for this, we are using the regionalized organoids and assembloids, and I'll tell you more about that in a few slides. And then we also try to correlate everything that we find in organoids with ex vivo human primary cortex, which is really the closest thing that we can get to where we can have live cells to work with and see if we can recapitulate some of these phenotypes or rescue compounds. So I primarily focused on cortical gray matter so far, and what I'm going to try to tell you about today are 3 projects that we've worked on, one during my clinical training and my research training, and 2 that are relatively new in my laboratory. And I'm going to start by telling you about the project focused on cortical astrocytes and how are those affected by hypoxia. That's the newest project in the lab. I'm going to tell you about the inhibitory neurons. And let me see if I can do this laser pointer. There, can you see it? Yes. About cortical inhibitory neurons and how are those affected by hypoxia, and how are cortical progenitors that are still proliferating affected by hypoxia. So briefly about the regionalized cortical organoid model, we published this in 2015. We're using induced pluripotent stem cells derived from fibroblasts, from control individuals, like to our knowledge, non-diseased individuals. We generated this model where we can take the iPSCs, put them in low attachment plates, and then by using specific growth factors, we tell them to become neural progenitors, and then we expand the neural progenitors, and later on with other specific growth factors, I will not go into the details of this, we are driving these cells to becoming neurons. And later on, if we keep them in culture long enough, they start forming astrocytes. This is how they look. They have, when we do immunostainings, we see that there is a ventricle-like area. Everything is like, nothing is like the actual brain. We have a ventricular zone rich in PAC6 radial glia progenitors. More superficially, there is a subventricular zone rich in TBR2 intermediate progenitors. And then more superficially, there are the neurons. So it resembles the developing human brain. This is a picture of an astrocyte. You can see in GFAP, they look in the 3D cultures, and then after we put them in monolayer, they don't have those reactive kind of morphology, which was really interesting to see. And then also we see neural network activity. So not only that the cells are there, but they're actually forming connections, and they connect each other they connect in networks. Separately, Sergiu Pașca's lab focused on generating regionalized subpallial organoids, and these primarily contain inhibitory neurons and inhibitory progenitors. And again, I'm not going to go into too much details here because this work was published in 2017, but practically they're showing that you can get NKX 2.1 progenitors, inhibitory progenitors, and then inhibitory neurons of multiple subtypes, somatostatin, calretinin, calbuildin, and parvalbumin, again, if you wait long enough in culture. Similarly, these cells are also connected in synaptic networks, and they respond to, they are inhibitory. So putting this together, this was very interesting. They also put this together, these organoids, where they connected cortical organoids that contain excitatory neurons and ganglionic eminence-like interneurons organoids, and they put this together in 4 brain assembloids, which really gives us the opportunity to study how the inhibitory neurons migrate during development. This is something that starts happening in the 2nd trimester of pregnancy, and in humans continues after birth. And there are a lot of neuropsychiatric disorders associated with abnormalities in the development or function of the inhibitory neurons. And I'll briefly tell you about that in the next slides. So with this model, we decided to focus on developmental hypoxic brain injury. Again, this is very important to me because a lot of the extremely preterm babies are exposed to these. because of lung immaturity and inability to really to transfer oxygen from the air into their lungs and then into their blood because their lungs are so immature. So first, I'm going to tell you about how does hypoxia affect synaptic pruning by astrocytes. I'm going to tell you about the migration of inhibitory neurons and how is this affected by hypoxia. And then also I'm going to talk, if I still have time, about the differentiation of TBR2 intermediate progenitors and how is this affected by hypoxia. So synaptic pruning by astrocytes. What do we know about astrocytes in cortical organoids? Well, there was a study published, led by Stephen Sloan in 2017, where they showed that in this type of regionalized organoids, after about 100 days, we start seeing astrogenesis in the organoids, and then they kept the cells up to 600 days, and they showed that they stabilize their numbers, but they do become more mature. As you can see here, between 180 and 320 days, their morphology changes. And then when they compared the transcriptome of primary astrocytes from human fetal brain and then postnatal brain up to like 60-something years, they found that, and they compared it with the astrocytes from the prenatal stages, they saw that the fetal astrocytes actually have a very different transcriptome compared to the ones from adults. And when they try to see what happens in organoids over time, after about 300 days, they were starting to see postnatal transcriptional signatures, which really demonstrated that indeed these cells do mature in vitro. And interestingly, they start having these post-transcriptional signatures after about 300 and something days, when it's about 10 months of pregnancy, which is term. So something tells them that they have to start changing their transcriptional signature without us really doing anything in culture. So this is the point that we focused on because this is relevant for the hypoxic ischemic encephalopathy. And what we did was we studied the effects of hypoxia on the synaptosome engulfment, because as I've shown you earlier, there is already synapse refinement at term equivalent. So what we found, I start with the conclusion so that you can focus on the data. We found that hypoxia does decrease the synaptosome engulfment by astrocytes, and then interestingly, this happens through disruption of the circadian rhythms in astrocytes, but not in neurons. So why do we care about synaptic pruning? Well, we know that there is an increased number of excitatory synapses in postmortem brains of individuals with autism spectrum disorders. We also know that there are circadian rhythm disruptions in ASD, so a lot of these patients suffer from sleep disorders. And then there are also polymorphisms in the circadian clock-related genes that are high risk for ASD. So with all of this in mind, we didn't know any of this except for the excitatory synapses in ASD when we started this project. We went on to see what happens when we expose astrocytes to hypoxia. So we grew organoids for 10 months in culture. This was work done by a fantastic postdoc in the lab, Lily. And after 10 months in culture, we dissociated the organoids. We fluorescently labeled astrocytes with GFAP, GFP. And then we exposed them to hypoxia for a total of 48 hours in monolayer. The first 24 hours, we did nothing. We just kept them in hypoxia. And in the last 24 hours, we added synaptosomes. And then we performed confocal live imaging. We validated that indeed we have hypoxia in these organoids and these cultures. And we looked to see, we looked at the synaptosome engulfment. And what we found was that under hypoxic conditions, there was a significantly decreased synaptosome engulfment by astrocytes, which was very interesting because until recently, people have primarily thought of microglia as being the primary synapse engulfment cell in the brain. So with this in mind, we went on and we performed RNA sequencing on isolating and purified astrocytes from control and hypoxic conditions from organoids that are over 300 days, because we wanted to see what are the transcriptional changes. We found that indeed there are some genes that are upregulated, some that are downregulated, but when we did geoanalysis, we found that we are seeing the expected things like cellular response to hypoxia, endoplasmic reticulum stress, glycolysis, but what we also found was that we saw changes in the circadian rhythm pathway, which was extremely interesting, and we also did not see this in RNA sequencing in neurons when we purified neurons. So we had no experience with circadian rhythms, and I think this is fascinating because we established a collaboration with Eric Gibson at Stanford, and then with Eric Herzog at WashU, and Eric sent us this PER2 luciferase construct where we can practically add this lentivirus on the cells and then look to see if we actually have circadian rhythms in the organoids. And we did this in control and hypoxia conditions, and what we found, which was really shocking to us, was that under control conditions, we indeed can see circadian rhythms in organoids, and these are disrupted by hypoxia. So again, this was really shocking to us. We were not expecting this, but it was very interesting to see how transcriptomics can actually teach us a lot of things, and then it also validates when you do these assays. So circadian rhythms. So I learned a lot about this, but I'm going to be very brief when I'm explaining. So practically, there is a feedback loop, and then the clock genes and the BMAL genes just come together, and practically when they are upregulated, they induce the production of the transcription of clock output genes. And then RevERB, for example, is an inhibitor. As soon as you start seeing a lot of clock output genes, there is a threshold, and at that point, RevERB, which is a master negative regulator of the circadian clock progression, starts getting upregulated and then inhibits the clock and BMAL. So we started looking into RevERB, again, as a master negative regulator of circadian rhythms. And the other reason why we looked into it is that because it was upregulated in the transcriptional data that we found in organoids. So we started looking at what happens with NR1D1, which is a gene for RevERB and clock, using RNA-scope. And we found that RevERB is indeed increased, transcription is indeed, mRNA, is indeed increased under hypoxic conditions, and then clock, which helps with the progression of the circadian clock, is decreased. We validated this at the protein level using Western blots, and then we quantified this, and indeed we showed the same thing with quantification. So next, we asked, how does this happen? Like, what are the phagocytosis genes that are associated with this defect in hypoxic astrocytes? And previously, there was a paper published by Chung when he was, he's now at KAIST Institute, but then when he did his postdoc with Ben Barres at Stanford, he found that these two phagocytosis receptors, MAGF10 and MERTK, are important for the phagocytosis properties of astrocytes. So what we did, we went back to the transcriptomics data, and we found that under hypoxic conditions, there was a decreased expression of MAGF10. However, there were no changes in MERTK. And we went on, and again, we validated this at the protein level. And I will not go too much into the details, but that's pretty much the quantification from multiple lines across multiple experiments. So what we found so far was that hypoxia induces circadian clock disruptions in astrocytes, but not neurons, and I haven't showed you the neurons data. But it also decreases the synaptosome engulfment by astrocytes. So the next question was, is there any direct relationship between circadian clock disruptions and synaptosome engulfment defects? And we went on to look into this by using a RevERB agonist. This was a pharmacological agonist. It's called SR9011. And we practically did the same experiment, but in the absence of hypoxia. And what we found was that indeed, when we add this RevERB agonist, there is less synaptosome engulfment by astrocytes. And then we looked at the MERTK and MAGF10, and then we saw again that while MAGF10 was, the expression of MAGF10 was decreased, MERTK was not affected at all. So Next, in collaboration with Professor Chung at KAIST, who studies circadian rhythms, we wanted to see if we can see this in vivo. And what his lab did was injecting this RevRB agonist into a mouse model, and then looked at the CA1 region. And what they found was that the same thing practically. They looked at astrocytes, and they found that when you add pharmacological activation of RevRB and inhibit the progression of the circadian rhythms and the clocks practically, you see a decrease in synaptosome engulfment by astrocytes within the hippocampus. And we looked at MAGEF10 again, and we see that the MAGEF10 is decreased under following the expression following the exposure of animals to this RevRB agonist. So with that in mind, with that, practically we had a full connection between, we know that hypoxia disrupts the circadian clocks, disrupts the synaptosome engulfment, but circadian clock disruptions alone can affect synaptosome engulfment by astrocytes. And this is really important because it's not only hypoxia that can be an environmental injury. We don't know what inflammation does. We don't know what a lot of medications do. So I think this just opens up a lot of questions. And one of the things that we are interested in looking at more is actually, do circadian rhythm disruptions lead to autism spectrum disorders or increase the risk of autism spectrum disorders through abnormal synaptic pruning? Because we know that ASD patients have extra excitatory synapses in the cortex. And the circadian rhythm disruption was just like seen as a clinical association. But are we actually starting this process of increasing the risk in the neonatal ICU by all of these injuries that babies are going through? So the next steps are to really study the developmental effects of circadian clock disruptions at a more global level. I actually have a narrow one that is being evaluated today. And then screening for neuroprotective compounds focused on circadian modulation, because there are a lot of those that are FDA approved, and the question is, can we start intervening sooner to prevent or decrease the risk for neurodevelopmental disorders in babies who are exposed to hypoxia as soon as the neonatal ICU. Next, I'm going to tell you about inhibitory neurons, as I promised. And this is also work that came out from my lab. My lab was opened in late 2018. So all of the work that I'm telling you about was practically done in about 2.5 years, because we were closed by the pandemic for a long time. And it just speaks to how amazing my team is, not me, my team. So inhibitory neuromigration, why are we interested in that? Well, it's pertinent to extremely preterm birth, to hypoxic ischemic encephalopathy, congenital heart disease, because as I told you before, these neurons, these inhibitory neurons, migrate from the precursor practically of the ganglionic eminences into the excitatory cortex to form connections and establish that excitation inhibition balance. And they represent about 20% of all cortical neurons in adults if everything goes well. And we noticed in post-mortem brains from extremely preterm babies, for example, that their numbers are reduced. And abnormalities in their development and function are associated with multiple neuropsychiatric disorders and are the cause for epilepsies. So again, I'll start with the conclusion. What we found using assembloids was that hypoxia decreases the migration of cortical inhibitory neurons or interneurons, and this is rescued by supplementation with adrenomedulin. And I'll tell you what adrenomedulin is, because again, the beauty of science, I didn't know what that was when we first started this project. So how did we start this? This project was led by Wojciech Migno, who was a postdoc in the lab, and he's a lecturer at Uppsala University now in Sweden, and Alisa Puno, who is an amazing research associate in my lab. And what they did was they used induced pluripotent stem cells. They generated assembloids, and using confocal live imaging, we fluorescently tagged the inhibitory neurons. And using confocal live imaging, they looked at the migration of these inhibitory neurons in control and hypoxic conditions for 24 hours. And they looked at pretty much at the same cells, like how does the same cell react to hypoxia, and compare that to the previous 24 hours when they were in control conditions. And the things that we looked at were the number of nuclear saltations. The inhibitory neurons migrate by saltating. It's very interesting, they jump. And then we looked at the saltation length and the directionality, because they have to move, they have to saltate to move, they have to jump a specific length, and then they also have to move in the right direction. And what we found was that under hypoxic conditions, there was decreased number of saltations in these inhibitory neurons, and we found no difference in saltation length. So when they jumped, when they saltated, they jumped the same length. And we also found no changes in directionality. So it was not like they didn't know what they were going. They were still going in the same direction. When we did transcriptional analysis, we found that the top differentially expressed gene, when looked at differences by log-fold chain, was this ADM, or adrenomedulin. And we validated this at the protein level. So indeed, when the transcriptional expression increases, the protein level also increases. And again, I didn't know what this was, so we started looking at adrenomedulin and its receptors. So adrenomedulin is a small 52 amino acid peptide, and it's more like an acute phase reactant under hypoxic and inflammatory conditions. It's being evaluated in sepsis as a marker of severity in adults. And there is a company actually that works on having an antibody that increases its biological activity to try to improve outcomes, clinical outcomes in patients with severe sepsis who are in the ICU. So ADM, we started looking into the literature. In order to function, it needs this disulfide bond, and it has 3 receptors, RAMP1, RAMP2, and RAMP3, and it seems like in most of the organs, RAMP2 is the receptor that is primarily responsible for its activity. It can function through multiple pathways, and then what it does, it increases the phosphorylation of Krebs, and then it increases the transcription of specific genes. So what we did, first, when we saw that it's so increased, like its expression is so increased, we performed RNA sequencing, single-cell RNA sequencing in control and hypoxic assembloids. So we practically exposed the assembloids to hypoxia, we cut them, and then we did single-cell RNA sequencing. And what we found was very interesting, I think. It seems like the progenitors, these are the subpallial, these are the pallial organoids, and we found that the progenitors, the astrocytes and the choroid plexus significantly increase the expression of adrenomedulin under hypoxic conditions, so not all cells. However, and then we looked at the RAMP2 receptors, and it seems that inhibitory and excitatory neurons both increase the expression of the receptors for adrenomedulin. And we found that RAMP1 is expressed, but it doesn't change under hypoxic conditions, and RAMP3 is not expressed at all in our model. Next, we wanted to see if, and this is going to be very counterintuitive, but we wanted to see if adding more adrenomedulin is going to help the cells migrate. And our thought behind this, because a lot of studies try to supplement things that are being downregulated, what we thought was, if the cells are trying so hard to make it, it has to be for a reason. So we added more adrenomedulin, exogenous adrenomedulin, and then we looked at the migration of inhibitory neurons. And what we found was that, again, under hypoxic conditions, there was a decrease in the saltation, but when we add and supplement the media with adrenomedulin at the time of hypoxia, the cells continue to migrate. And here we looked also at the saltation length, and we saw no changes. Importantly, and I'm not showing that, we saw that under control conditions, when we just add adrenomedulin to control organoids, it has and inhibitory neurons, it has no effect on the saltation. So if everything is normal, it doesn't do anything. Next, we went and validated this in human fetal tissue. So we took slices of human fetal tissue at around 20 weeks, I think, for this project. And then we fluorescently labeled the same type of migrating inhibitory neurons, and we found that indeed adrenomedulin is also increased under hypoxic conditions. And then when we add extra adrenomedulin to the slices of human fetal tissue, we see that there is a rescue of the migration, but no change in saltation length or directionality. So again, if cells secrete ADM by themselves, why add more? And what we know is that under physiologic conditions, I told you about this disulfide bond. This one is really important, and it happens, the formation of this happens in the endoplasmic reticulum, which is functional under physiologic conditions, and then this disulfide bond is crucial for the binding to the receptor. So looking at the literature, we found that hypoxia actually inhibits the formation of this disulfide bond. This disulfide bond formation is oxygen dependent and is also dependent on functional ER, which both are affected under hypoxic conditions, and as a lot of the literature is showing. So practically what we think that is happening is that the cells are making adrenomedulin, which we've demonstrated, and the literature is demonstrating that for other conditions outside of the brain. But we think that this binding to the receptor is impaired, and thus the biological activity is decreased. So when we add exogenous adrenomedulin that has a disulfide bond, then this binds to the receptor and helps with the downstream pathway activation. So making another long story short, practically what we've shown is that when you add exogenous adrenomedulin, it binds to the receptors under hypoxic conditions, it activates the cyclic AMP-PK pathway, increases the phosphorylation of Krebs, and then it also increases the transcription of the GABA receptors. And previous literature has shown that GABA receptors, the expression of GABA receptors is related to the migration. So we think that this is I'm not even saying like the main, it's one of the mechanisms by which adrenomedulin helps the cells migrate under hypoxic conditions. And last but not least, I'm going to tell you about the first project that I actually did. This was still done during my training, where we looked at the defects in generation of cortical excitatory neurons following exposure to hypoxia. And again, this is pertinent to preterm brain injury, to congenital heart disease for babies who are already hypoxic in utero because of abnormal mixing of the blood in the heart. And we know that both extremely preterm babies and babies with congenital heart disease, specific congenital heart diseases, have simplified gyri. They have a lower cortical thickness in some areas of the brain. There is increased water content, less arborization of the neurons, and then there is obvious increased risk for neurodevelopmental disorders. And again, the conclusion, hypoxia induces premature neuronal differentiation of TBR2 intermediate progenitors, and this is rescued by ISRIB, and I'll tell you briefly about ISRIB. How did we do this? We took organoids at around 80 days when they contain progenitors, they contain intermediate progenitors and some neurons, and we wanted to see what happens when we expose them to hypoxia. We took this extra step, so I know there it says that we're doing less than 1% oxygen. This is how we set the equipment to. However, we showed that the equipments are not perfect. And then when we measure the oxygen level by using a sensor, an oxygen sensor, we saw that under control conditions, our partial pressures of oxygen were around 80. And then when we exposed them to hypoxia, they were around 25, 30. And this was enough to induce hypoxia at the protein level by stabilization of the HIF-1 alpha protein. Again, we did transcriptional analysis. We saw a lot of differentially expressed genes at 24, 48 hours, nothing at 72 hours after we reoxygenated the cells. But then when we looked at the cells that are primarily sensitive to hypoxia, we found that the ones in the subventricular zone are the one, compared to all the other cells, they seem to be primarily affected by hypoxia. And the reason why this is important is because the subventricular zone and the outer subventricular zone is highly expanded in primates during development and during the fetal life compared to any other animals. And we think that this outer subventricular zone is responsible for the expansion of the human brain and the primate brain. So this would be a very difficult phenotype to capture in a rodent model, for example. So this was very interesting, and then we went on to actually count the number of TBR2 intermediate progenitors in control versus hypoxic conditions. And we found that indeed under hypoxic conditions there were less TBR2 intermediate progenitors, and this has not, their number has not recovered 3 days after reoxygenation. We found no difference in the PAC6 radial glia progenitors, suggesting that indeed there might be a different, like maybe an evolutionary difference in how these cells respond to hypoxia. From the transcriptional data, we found that endoplasmic reticulum stress, practically. And then again, I didn't know about the endoplasmic reticulum stress. It was primarily worked on in cancer previously. It's now being studied more and more in development and neurodegenerative diseases. But practically, when you have endoplasmic reticulum stress, for whatever reason, you have the upregulation and the activation of the unfolded protein response. And this, under limited stress, it promotes survival of the cells. But when the stress is too prolonged or too severe, it leads to apoptosis. We found that indeed, under hypoxic conditions, more cells were expressing this ATF4, which is a marker of the of unfolded protein response. And we saw that under when we add ISRIB, which is a blocker of one of the arms of unfolded protein response, this rescues the number of TBR2 intermediate progenitors under hypoxic conditions. So first we thought that the cells are dying and that's why they're not recovering. We found no cell death, actually. And then we started looking at the cell cycle. And we know from the literature that this P27 protein, when expressed, it blocks the transition from G1 to S of the cell cycle. And then if this lasts for very long, for a specific amount of time, then the cells start exiting the cell cycle. And when we looked at the expression of this protein, we found that under hypoxic conditions, this is significantly upregulated in hypoxic conditions and is rescued by ISRIB. And then we also found that under hypoxic conditions, the cells are starting to express CTIP2, which is a marker of early neurons, but this is also rescued by hypoxia. So practically, they are exiting the cell cycle. They are not dying, they're just exiting the cell cycle. However, this is very important because if you have progenitors that are supposed to symmetrically divide and make new progenitors, then you could imagine that long term, you are decreasing the total number of neurons. And this could be, again, one of the mechanisms by which the brains of extremely preterm babies, congenital heart disease babies, are in some areas of the brain smaller. We validated this in human fetal tissue by quantifying the TBR2 and PAC6 progenitors, and we found that, again, indeed, TBR2 intermediate progenitors are decreased, rescued by ISRIB. So these are the 3 projects that I really wanted to tell you about today, and I don't think I have to convince you, because this is a stem cell institute, but that human cellular models are really useful to try to understand the molecular mechanisms of brain injury and recovery. They complement in vivo animal studies, and they might be able to help us identify interspecies differences. And they're a great tool for screening for neuroprotectives. And this is something that we are doing in our lab using, we can screen thousands of organoids at the same time right now. And with that, I would like to thank you all for being here and for inviting me to talk about our work. And to my lab, they are all amazing, and we like to work hard, but we like food and traveling to Disney. Thank you to all of our collaborators and our funding agencies. Thank you again, and I'll take questions.
Thank you very much for a very nice talk. So you showed really nicely how hypoxia and synaptosome engulfment and the circadian clock are connected. I'm wondering whether if you remove the hypoxia component, do you think that disruption of circadian clock by itself also cause defects in synaptic engulfment? Because it could be relevant to other conditions. Right?
Yeah. Sorry, I showed that. Maybe I didn't emphasize it enough. Yes, so we did that. We took out hypoxia, and we wanted to see if there is a direct connection between circadian clock dysregulation and synaptosome engulfment. And we did that by pharmacological induction of RevERB, and we are seeing the same thing. So you don't need hypoxia. I think any injury that affects the circadian rhythms and the circadian clocks can lead to the same thing. And just circadian rhythm abnormalities and disruptions by itself can cause that. And that's why I'm so worried, because we have a lot of kids, for example, with obstructive sleep apnea. We have a lot of other children who suffer from circadian rhythm disruptions, and we know that all of these disorders are associated with neurodevelopmental impairments. So I don't think it's just a neonatal or like preterm baby injury kind of effect. It can also affect older children who are still going through synapse refinement for a long period of time. Yeah, thank you for the question.
There's a question way down there.
Great talk, thank you. I was curious if you looked into mitochondrial dysfunction and the hypoxic conditions, I mean, with the Ross species, or even if varying levels of hypoxia, like different lengths of time, have impacted mitochondria even further down the line?
Yes, that's a great question as well. And we are looking into the mitochondrial dysfunction. We're seeing that the mitochondria is fragmented as expected. We are doing seahorse analysis on this, and it actually is really beautiful. The other thing that we are studying mitochondria in is 22q11 deletion syndrome, where again we see dysfunction in mitochondria, which is very, it's really fascinating, and we're seeing it in the human fetal tissue as well. So we think that there is actually a direct, there is a common pathway of injury between hypoxia and patients with 22q11 deletion syndrome, which really have the highest risk for neuropsychiatric disorders. There's like a 20-fold increase in neuropsychiatric diseases. So yes, we are looking at mitochondria. They're beautiful. And one of my postdocs actually developed a ton of tools to look at, to visualize mitochondria, to visualize ROS production, to visualize the membrane potential, and to see how it interacts with the ER, for example. We're also looking at how astrocytes and neurons, because there was that beautiful paper where astrocytes transfer mitochondria to neurons, and now we're looking at how are astrocytes actually transferring mitochondria to neurons under hypoxic conditions, and how does that happen if it does, and can we make it better if they don't? So yeah, I love mitochondria. I could talk about it forever.
Obviously, when we use the term hypoxia for a fetus is normoxia.
Right.
So whenever we're doing cultures, we're not entirely mimicking. In fact, when we go down to, let's say, 5% or 8%, we're mimicking what is normal for fetal brain development. How do you parse out what should actually be a normal response in utero versus what truly is a pathologic response to a pathologic oxygen tension?
Right. That's a great question as well. And I've been thinking about this because this was one of the first questions I think that we started getting when we published this, or we started talking about it. I think in vivo versus in vitro is different. So in vivo, you have, the cells are not hypoxic, like even at partial pressures of oxygen of 25 millimeters of mercury, the cells in the fetal brain do not express markers of hypoxia, right? And then, so the other, and I kept thinking about this, and one of the major things is that in utero you have fetal hemoglobin. And fetal hemoglobin, what it does, it captures significantly more oxygen from the environment, and it releases it to the cells in a more controlled way. So if the cells are asking for it, it's going to give it because of the dissociation curve. So I think in vivo we are not seeing hypoxia despite, like we are in a maybe hypoxemic environment, but the cells are not hypoxic, because I think of the fetal hemoglobin being one of the major factors. So what we can do in vitro is mimic what the cells are doing when they're not hypoxic versus what the cells are doing when are hypoxic. And we think that this is relevant because, again, fetal cells are not hypoxic even at 25 to 30. So that's how I'm thinking about it.
And I notice you're using a very, you're actually going to less than 1%.
Less than 1%, but as I showed, because we actually measured this, because 1% should be around like 7 millimeters of mercury, so we measured this, and actually the equipment, like yes, the equipment, we set it at less than 1. However, when we do the oxygen measurements, they are like 30. So even partial pressure of oxygen of 30 are enough to induce to stabilize HIF-1 alpha in our system. And we think that that's what we are comparing. We're comparing control, like non-hypoxic with hypoxic cells.
You got complimented on a fantastic talk, and you mentioned that hypoxia affects the cell cycle. So do you think that hypoxia is beneficial for impacting tumors?
Affects, well, not in cancer cells, because cancer cells don't. So neurons primarily need they need mitochondria, and they need oxygen for the mitochondria to function. And I don't think, like we know that. We know that this is why hypoxia affects neurons. It's like you have strokes, you have all kind of diseases associated with hypoxic injury. So neurons and brain are highly dependent on mitochondria to function in order to survive. Cancer cells are primarily glycolytic. They don't rely on mitochondria as much. So I think that's why they can bypass. They don't care about hypoxia as much, because they are glycolytic. They produce their ATP in the first steps of glycolysis, not through oxidative phosphorylation, through mitochondria. So I think that's where the big difference is. And there are a lot of studies that look at this and like chemotherapy-related compounds targeting HIF-1 alpha, because the goal is to prevent that, if I remember correctly.
Okay. Any questions in the room? Okay. This actually is coming from the Winchell Boris lab at Case Western. And very interesting results, and thanks for sharing. Specifically for the progenitors that were exiting the cell cycle before completing the number of symmetric divisions required, they indicate that they saw similar mechanisms in DNA repair defects related to microcephaly, for example, LIG4. They're wondering whether you have any insights on the level of DNA damage caused by hypoxia in this mechanism by using your system.
Yeah, that's a great question, and I have not looked specifically at the DNA damage. We can definitely look into that. There's probably some, but I cannot say that it is. We do know that various levels of hypoxia induce various severity of cell damage. So for example, the 25 to 30 millimeters of mercury induce cell stress, but not cell death, as we've shown. We have a model where we have 5 millimeters of mercury, and there we start seeing actual cell death. So I think there are like various degrees of insults that you can get in this model. But we can definitely look at the DNA damage. Yeah, that's a great point.
Questions in the room? If not, I'll go back to the Zoom. This questioner wanted to know, you've talked a lot about the role of hypoxia in development or aberrant development. Can you speculate about the role of perfusion, lack of perfusion, which is also the second part of?
Yes, absolutely. So perfusion, I think it's more relevant for term hypoxic encephalopathy, because we know, again, there can be cord compression, there can be placental detachment and placental insufficiency. I don't know if it's as relevant for extremely preterm babies, because we can control the blood pressures there, but we still have hypoxic events because of the significant lung immaturity. We can literally not get oxygen through the lungs to get to the tissue. So yes, reperfusion can be important, but we do know that the reperfusion can actually cause injury because of the reoxygenation stress, unless you also have hypoglycemia, because the brain cells really need glucose and they need oxygen to function. So if you have prolonged ischemia as well associated with hypoxia, then I think you can deprive the cells of glucose as well, and that can make things significantly worse. However, it is well known in the literature that reoxygenation during reperfusion is one of the main reasons, because it increases the oxygen species, and reactive oxygen species you have majorly affected mitochondria that doesn't know what to do with the oxygen. So yes, it's important for some of the diseases, I think, but also through the reoxygenation primarily.
Okay.
There is a question in the room.
Oh.
What led to the premature 24-week baby? Was it an acute event or a chronic event?
From what I remember, so there are multiple things. Sometimes the majority of women go into preterm labor and we don't know why. They don't seem to have an infection. They don't seem to have placental insufficiency. We just don't know why they go into preterm labor and deliver. And then for some cases, we know that there is placental insufficiency, and then the babies just don't grow in utero, and then at some point, the body just decides that, okay, like it's not doable anymore. So then they go into preterm labor. One of the other causes is premature rupture of membranes. So for some reason, the membranes rupture, and then women go into preterm labor, and then infection is another major cause of preterm labor. But for the most part, we don't know what induces preterm labor. We think that most of the time it has to do something with the placenta that we are still trying to figure out. Yeah.
You've tracked, you've used HIF-1 alpha to mark the degree of hypoxia. Does HIF-1 alpha itself play any role in either aberrant or normal development in your system?
Well, we know that it's the first step of activating the hypoxia responsive genes pathway. We have not specifically looked at directly at like HIF-1 alpha binding to these to specific. We haven't looked at HIF-1 alpha, to be honest, like directly. It's just we see it as a marker of hypoxia, and it's always there. It's just that it gets stabilized under hypoxic conditions, and then it starts the cascade of hypoxic, activating the molecular pathways for hypoxia, which we think are inducing the endoplasmic reticulum stress and mitochondrial dysfunction and metabolic changes, of course.
Great. Any other questions in the room? There's maybe two last ones on Zoom. Well, I'll do the two ones on Zoom, and you can think about whether you have any questions here. One question deals with, you found very interesting results in conjunction with congenital heart disease. Your system is very well designed to look at genetic effects on development. Do you think there's any possibility that the cerebral abnormalities are not necessarily due to epigenetic events like hypoxia, but that the same genes that cause congenital heart disease could also be causing cerebral abnormalities?
Yeah, that's a great question, a fantastic question. We're actually working with Casey Gifford at Stanford. She's an assistant professor in cardiology, and she's very interested in the connection between congenital heart disease and brain abnormalities. So we have a collaborative project working on this to try to understand how genes previously associated with congenital heart disease, or not even yet associated with congenital heart disease, can impact the development. Yes, so we're working on that.
Okay. Any other in the room questions before I do the last Zoom question? Well, this is a good last question because it's fairly open-ended. It sounds as if you believe that astrocytes have not been receiving the attention they deserve in brain development, and that microglia have been receiving it all, but that astrocytes should get more respect. What do you think?
I completely agree with that. And I think when you think about development, like microglia are there from the beginning. Astrocytes start increasing their numbers in the second trimester of pregnancy, and when you think about it again in terms of numbers, microglia always stay at around 5%, and then astrocytes increase to be like, I don't know, 40%, depending on the brain region, in terms of the proportion of cells. So I do strongly think that we should start looking more into astrocytes and their role in synaptic pruning. And there are a few labs who are interested in this. So yes, we're going to make progress. Keep working on astrocytes.
Well, with that, thanks so much, Anca. And we'll see you all for the April installment of the Southern California Stem Cell Consortium.